Hello students, myself Sarita Devi, Biology lecturer, Government Girls Senior Secondary School, Jandiala. Today we will study 5th chapter of 12th class Biology, that is Principles of Inheritance and Variation, and topics are Chromosomal Theory of Inheritance, Linkage, Types of Linkage, Crossing Over and Recombination, Mechanism of Crossing Over, and Chromosomal Maps. So, chromosomal theory of inheritance was given by Walter Sutton and Boveri in 1902. So, these two workers found a close similarity between the transmission of hereditary characters and the behavior of chromosomes while passing from one generation to next through agency of gametes. So first of all, we should know what are chromosomes. So chromosomes, chromo word means colored and somes mean body. So they are the colored body and they are present in the cell inside the nucleus. And they are thread-like structures which are made up of nucleic acids and proteins, and they are mainly involved in carrying genetic information in the form of genes. So in each body cell there are two types of cells that are diploid as well as haploid. In case of diploid cells, which are the normal body cells, the number of chromosomes in humans is 46, and in case of haploid cells, which are mainly sperms and ova, these are the gametes, the number of chromosomes is 23. And the term inheritance means transmission of characters from parents to progeny. So here we have to study how chromosomes are inherited from parents to offspring. So in 1865, Mendel gave law of inheritance and law of independent assortment, but his work was published, but his work was not recognized till 1900. ਸੋ ਕਈ ਕਾਰਨ ਸੀ ਜਿਸ ਕਾਰਨ ਉਹਨਾਂ ਦੇ ਮੈਂਡਲ ਦੇ ਵਰਕ ਵਾਸ ਨੋਟ ਅਸੈਪਟਡ. First reason was that communication was not easy at that time, and moreover his work was not widely published like ਹੁਣ ਦੇ ਸਮੇਂ ਆਪਣੇ ਕੋਲ YouTube ਹੈ, Facebook ਹੈ. At that time the work was not published. Secondly, jada Mendel the character was that genes are discrete units and stable, or and alleles, they do not blend with each other. And his findings that was not accepted, because they observed that in nature there is a continuous variation in nature. So ਅਸੀਂ ਕਹਿ ਸਕਦੇ ਹਾਂ ਕਿ ਮੈਂਡਲ ਦੇ ਅਕੋਰਡਿੰਗ ਜਿਹੜੀਆਂ ਜੀਨਸ ਹੈ ਉਹ ਡਿਸਕ੍ਰੀਟ ਯੂਨਿਟ ਸੀ. ਡਿਸਕ੍ਰੀਟ ਦਾ ਮਤਲਬ ਹੈ ਕਿ ਪਾਰਟ ਆਫ ਸਮਥਿੰਗ ਲਾਰਜ, ਕਿਸੇ ਵੱਡੀ ਚੀਜ਼ ਦਾ ਛੋਟਾ ਹਿੱਸਾ. ਸੋ ਉਹਦੇ ਅਕੋਰਡਿੰਗ ਮੈਂਡਲ ਦੇ ਕਿ ਜੀਨਸ ਹੈ ਜਿਹੜੀਆਂ ਉਹ ਸਟੇਬਲ ਹੈ ਐਂਡ ਦੇ ਡੂ ਨਾਟ ਚੇਂਜ ਫਰੋਮ ਬਰਥ ਟੂ ਡੈਥ. ਐਂਡ ਜਿਹੜੀਆਂ ਅਲੀਲਸ ਹੈ, ਅਲੀਲੋਮੋਰਫਿਕ ਪੇਅਰਸ ਹੈ ਜਿਹੜੇ ਜੀਨਸ ਦੇ, ਔਰ ਉਹ ਵੀ ਇੱਕ ਦੂਜੇ ਨਾਲ ਬਲੈਂਡ ਨਹੀਂ ਕਰਦੀਆਂ. ਬਟ ਜਿਹੜੇ ਕੰਟੈਂਪੋਰੇਰੀ ਸੀ ਮੈਂਡਲ ਦੇ ਓਬਜ਼ਰਵ ਦੈਟ ਕਿ ਨੇਚਰ ਵਿੱਚ ਜਿਹੜੀਆਂ ਕੰਟੀਨਿਊਸ ਵੇਰੀਏਸ਼ਨਸ ਹੁੰਦੀਆਂ. ਉਸ ਟਾਈਮ 'ਤੇ ਜਿਹੜਾ ਮੈਂਡਲ ਯੂਜ਼ਡ ਮੈਥਮੈਟੀਕਲ ਫਾਰਮੂਲਾਸ ਇਨ ਬਾਇਓਲੋਜੀ ਫॉर द फर्स्ट टाइम. Scientists did not accept mathematics in biology. So it was not clear कि genes where the genes are present in the body और किस material दियाँ वो genes बनियाँ हैं. So no importance was given to Mendel's work. So where are the genes present in the body or cell? It was not explained. And from which material are the genes made up of? That was not explained by Mendel. So no importance was given to Mendelian work. But in 1900, three scientists, De Vries, Correns, Teschmack, rediscovered Mendelian work और उन्होंने भी same results मिले. And Sutton and Boveri rediscovered and observed the behavior of the chromosomes when the cells were dividing. So ਜਦੋਂ cells divide ਕਰਦੇ ਹੈ ਤੇ ਉਸ ਟਾਈਮ ਤੇ ਉਹਨਾਂ ਨੇ chromosomal behavior note ਕੀਤਾ ਔਰ due to the advancement in the microscopy ਉਸ ਟਾਈਮ ਤੇ ਅਵੇਲੇਬਲ ਸੀ ਔਰ ਉਹਨਾਂ ਦਾ ਟਾਸਕ ਜਾਂ ਕੰਮ ਕਰਨਾ ਬਹੁਤ ਸੌਖਾ ਹੋ ਗਿਆ. So they proved that Mendel's law with the help of chromosomal movements. They showed the segregation of chromosomes during meiosis in cell division. और उन्होंने ਜਿਹੜਾ कि they led to the discovery of structures in the nucleus. And ਇਹ structures ਜਿਹੜੇ nucleus ਦੇ ਅੰਦਰ ਹੈ they got double and ਦੇ ਨੰਬਰ ਜਿਹੜਾ ਸੀ ਉਹਨਾਂ ਦਾ ਡਬਲ ਹੋ ਜਾਂਦਾ ਸੀ ਬਿਫੋਰ ਡਿਵੀਜ਼ਨ or during the cell division. So they proved Mendel's law with the help of chromosomal movements. And also they show the segregation of chromosome during meiosis cell division. So ਜਿਹੜਾ idea ਸੀ chromosomal segregation combined with Mendelian principle gave rise to chromosomal theory of inheritance. Chromosomal theory of inheritance is the fundamental theory of genetics thi. So according to this theory, genes are units of heredity and they are found in chromosomes. So here chromosomes and genes, they occur in pairs. Two alleles of a gene pair are located on the homologous chromosome. So these are homologous chromosomes. Or here there is a locus for flower color gene. This is allele for purple and this is allele for white. So हर किसी character या trait ਲਈ दो allelomorphic pairs है for that particular trait. और जिस location ते allele है that is known as gene locus. So let us study chromosomal theory of inheritance in detail. It states that genes are located at specific loci on the chromosome. Loci means the place where the genes are located on the chromosome. And it is the chromosomes which segregate and assort independently during meiosis, that is a cell division, and they recombine at the time of fertilization in the formation of zygote. So what are the features of chromosomal theory of inheritance? First is gametes, where the number of chromosomes is haploid. They serve as bridge between one generation and next generation, or they are the hereditary material. Secondly, both the sperm and the egg, they contribute equally in the heredity of the offspring. The sperm provides the nuclear part to the zygote. and there is a fusion of the sperm and egg nuclei during the fertilization to restore the same number of chromosomes. Now next is nucleus contains chromosomes. So if this is a cell, this is the nucleus and these are the chromosomes. So nucleus contains the chromosomes. Therefore chromosomes must carry the hereditary traits. Every chromosome or chromosomal pair has definite role in the development of an individual. If there is a loss of complete or a part of a chromosome, it produces structural and functional deficiency in the organism. Next is, like the hereditary traits, chromosome retains their number, structure, individuality throughout the life of an organism and from generation to generation. Both the chromosomes as well as the genes, they occur in pairs in somatic or diploid cells. So these are a pair of chromosomes, homologous pair of chromosomes, and these are the sister chromatids, and these are the non-sister chromatids, and their chromosome is connected with the centromere. So both the chromosomes as well as the genes, they occur in pairs. A gamete contains only one chromosome of a type and only one of the two alleles of a character. The paired condition of both chromosomes as well as the genes is restored during fertilization. Homologous chromosomes synapse during meiosis and then they segregate independently into the different cells. So lastly, in many organisms, sex of an individual is determined by the specific chromosomals. They are called the sex chromosomes. So this is a human karyotype. This is a karyotype of male and this is a karyotype of the female. So here 22 pairs are of autosomes which are similar, and 23rd pair, that is the sex chromosome. In case of the male it is XY, in case of the female it is XX. So this completes chromosomal theory of inheritance. Mendel said genes are stable, but Thomas Hunt Morgan helped to discover the basis for variation that occur after sexual reproduction. So sexual reproduction ਤੋਂ ਬਾਅਦ variations ਆਉਂਦੀਆਂ ਹੈ organisms ਦੇ ਵਿੱਚ। experimentally prove ਕੀਤਾ ਗਿਆ by Morgan। ਸੋ ਜੋ chromosomal theory of inheritance ਸੀ ਉਸ ਨੂੰ experimentally verify ਕੀਤਾ ਗਿਆ by Morgan after he conducted experiments on Drosophila melanogaster fruit fly ਤੇ। Thomas Hunt Morgan was an American genetist और उन्होंने Nobel Prize मिला in 1933. So he is considered as father of experimental genetics for his work on discovery of linkage kitty and crossing over, sex linkage and crisscross inheritance. So linkage was first discovered by Batson and Punnett in 1906 in Sweet Pea Lathyrus odoratus, but it was Morgan जिन्हों ने clearly prove किया और define किया linkage on the basis of experiments that were conducted on fruit fly. The term linkage was coined by Morgan. So now let us discuss the chromosomal theory of linkage. So here the linked genes, they occur in the same chromosome. If this is one chromosome, here are the genes, they are closely linked to each other. So this is A, B, C, D, and these genes, they are closely related to one another, and they are known as linked genes, and they occur in the same chromosome. And these genes, they lie in a linear sequence on the chromosomes. So there is a tendency to maintain the parental combination of genes in linked genes. So the linked genes, they always maintain the parental combination of genes. The strength of the linkage between the two genes, it is inversely proportional to the distance between the two genes. So if the genes, they are very close to each other, then the strength of linkage will be more. If the distance is more, then there are chances that the two genes may not stay together. So the two linked genes show high frequency of crossing over if the distance between them is higher. Now these two genes, they will show high frequency of crossing over if the distance between them is higher, and lower frequency if the distance is small. So there is less or lower frequency of crossing over if the genes are closely linked to each other. Now, what are linkage groups? Genes that are present on the same chromosome, they make one linkage group. They inherit together except for crossing over. Two homologous chromosomes possess either similar or allelic genes on the same loci, they constitute the same linkage group. सो अगर दो होमोलोगस क्रोमोसोम्स है और ਉਹਨਾਂ ਦੇ ਆਈਦਰ ਉਹਨਾਂ ਦੇ ਉੱਪਰ ਸੇਮ ਜਾਂ ਅਲੀਲਿਕ ਜੀਨਸ ਹੈ ਔਨ ਦਾ ਸੇਮ ਲੋਕਸ ਜਿੱਥੇ ਜੀਨਸ ਪ੍ਰੈਜ਼ੈਂਟ ਹੁੰਦੀਆਂ ਉਹਨਾਂ ਦਾ ਇੱਕ ਸੇਮ ਲਿੰਕੇਜ ਗਰੁੱਪ ਹੋਏਗਾ. The number of linkage groups present in an individual corresponds to the number of chromosome in one set of its genome. So humans ਦੇ ਵਿੱਚ 23 pairs of chromosomes ਹੈ ਤੇ linkage group ਹੋ ਗਏ 23 linkage groups. Now what is the significance of linkage? It reduces the chances of formation of new combination of genes in the gamete. So it helps to keep the parental, racial and important traits together. ਜਿਹੜੇ ਚਾਂਸਿਸ ਹੁੰਦੇ ਆ ਨਵੇਂ ਕੋਂਬੀਨੇਸ਼ਨਸ ਬਣਦੇ ਦੈਟ ਆਰ ਰਿਡਿਊਸਡ ਡਿਊ ਟੂ ਲਿੰਕੇਜ ਔਰ ਜਿਹੜਾ ਪੈਰੈਂਟਲ ਕੋਂਬੀਨੇਸ਼ਨਸ ਹੈ ਦੈਟ ਆਰ ਰਿਟੇਨਡ. ਲਿੰਕੇਜ ਜਿਹੜੀ ਹੈ ਡਿਸਅਲਾਉ ਕਰਦੀ ਹੈ ਪਲਾਂਟ ਬਰੀਡਰਸ ਜਾਂ ਐਨੀਮਲ ਬਰੀਡਰਸ ਨੂੰ ਟੂ ਕੰਬਾਈਨ 올 ਵੈਲਿਊਬਲ ਟ੍ਰੇਟਸ ਇਨ ਅ ਸਿੰਗਲ ਵਰਾਇਟੀ. ਲਿੰਕੇਜ इज़ अनडिज़ायरेबल और अनडिज़ायरेबल मनी जाती है अगर when desirable और undesirable genes they are linked together because ਉਹਨਾਂ ਨੂੰ separate नहीं किया जा सकता. Now let us see what is linkage and recombination. So linkage, it is the tendency of genes to stay together during inheritance through generations without any change or separation due to the presence on the same chromosome. So one gene and two gene, they are very closely related to one another or the ਇਹਨਾਂ ਦੇ ਵਿੱਚ chances separation ਹੋਣ ਦੇ ਬਹੁਤ ਹੀ ਘੱਟ ਹੈ. Genes that are located in the same chromosome are called linked genes. So a linked genes ਹੋ ਗਈਆਂ and those present in different chromosomes that are known as unlinked genes. So these two are not linked genes and these two are not linked genes. So linked genes, these genes occur on the same chromosome and do not show independent assortment, but they remain together. So jada dihybrid cross ratio, that is 3:1, jada like monohybrid ratio jo, and test cross ratio of 1:1. Progeny consists of only parental types, recombinants are absent. Unlinked genes, they occur on the different chromosomes and they show independent assortment. Dihybrid ratio is 9:3 is to 3:1, while dihybrid test ratio is 1:1 is to 1 in both the crosses. 50% are parental type and 50% are the recombinants are formed. So let us see the difference between the linked genes and unlinked genes. Genes that are placed on the same chromosomes and do not show independent assortment at the time of gamete formation, they are known as linked genes. The genes that are located on the different chromosomes and undergo independent assortment, or they separate or they segregate, that are known as unlinked genes. Linked genes show dihybrid ratio of 3:1, whereas the dihybrid ratio of unlinked genes is 9:3:1. In dihybrid, they show test cross ratio of 1:1, whereas in dihybrid they show test ratio of 1:1:1. Now, what are the characteristics of linkage? Linkage involves two or more genes which are located in the same chromosome. It reduces the chances of variability. Linkage may involve either dominant or recessive alleles. Linkage involves those genes which are located close to each other. जितनी ज़्यादा strength of linkage होएगी, it depends upon the distance between the linked genes. Lesser the distance, higher the strength, and vice versa. Types of linkage, ਜਾਂ ਤੇ complete linkage ਹੋ ਸਕਦੀ ਹੈ ਜਾਂ ਫਿਰ incomplete linkage. Complete linkage means in case of Drosophila and females of silkworm, there is a complete absence of recombinant types due to absence of crossing over. Incomplete linkage ਦੇ ਵਿੱਚ if some frequency of crossing over occurs between the linked genes, it is known as incomplete linkage. It is mostly seen in Drosophila female and P, or in case of maize. So complete and incomplete linkage, it is based upon the type of crossing over. Complete linkage, when the genes they are very close to each other and they show complete linkage. Genes are at some distance, or in case the chromosomes have tendency to separate. So in complete linkage there is no crossing over, and in incomplete linkage there is crossing over. Now, types of linkage based on the chromosomes involved. So based on the chromosomes involved, it may be autosomal linkage or sex linkage. So it refers to linkage of those genes which are located on the autosomes, means other than the sex chromosomes. Sex linkage means if the genes they are located in the sex chromosomes, that is X or Y. So next topic is crossing over. Crossing over means when the linked genes they don't stay together. The linkage groups can be separated by crossing over during prophase 1 of meiosis. There is a random exchange of DNA between the two non-sister chromatids of homologous chromosomes. is known as crossing over. So here these are a pair of homologous chromosomes, and there is crossing over between the two non-sister chromatids of homologous chromosomes. So here this is non-recombinant chromosome, and this is also non-recombinant chromosome, and these are recombinant chromosomes, or where crossing has taken place. So what are the major steps in meiotic crossing over? So meiosis ਦੇ ਵਿੱਚ ਜਿਹੜੀਆਂ 4 steps ਹੈ. First is synapsis, then second is duplication of chromosomes, then crossing over, then terminalization. So synapsis ਵਿੱਚ pairing of homologous chromosomes ਹੁੰਦੀ ਹੈ, and they form bivalents. Then there is duplication of chromosomes. Homologous ਜਿਹੜੇ chromosomes ਹੈ, they get duplicated to form chromatids. So 4 chromatids formed है. Then there is crossing over, that means exchange of chromosomal segments between the two non-sister chromatids. So ਇਹ ਦੋ ਜਿਹੜੀਆਂ center ਦੇ ਵਿੱਚ ਹੈ, these are non-sister chromatids. There is exchange of genetic material. ਲਾਸਟ ਸਟੈਪ ਦੇ ਵਿੱਚ ਟਰਮੀਨਲਾਈਜੇਸ਼ਨ, ਜਿਹਦੇ ਵਿੱਚ ਸੈਪਰੇਸ਼ਨ ਹੋ ਜਾਂਦੀ ਹੈ ਨੋਨ ਹੋਮੋਲੋਗਸ ਕ੍ਰੋਮੋਸੋਮਸ ਆਫਟਰ ਕ੍ਰੋਸਿੰਗ ਓਵਰ. ਸੋ ਇਹਦੇ ਵਿੱਚ ਰੀਕੋਂਬੀਨੈਂਟ ਕ੍ਰੋਮੈਟਿਡਸ ਦੀ ਫੋਰਮੇਸ਼ਨ ਹੁੰਦੀ ਹੈ. ਨਾਓ ਕੈਰੈਕਟਰਿਸਟਿਕਸ ਆਫ ਕ੍ਰੋਸਿੰਗ ਓਵਰ. First it occurs at 2 levels, chromosomal level ਜਾਂ DNA level ਤੇ crossing over ਹੁੰਦੀ ਹੈ. Crossing over occurs between the non-sister chromatids of the homologous chromosomes. ਜਿਹੜੀ exchange of genetic material is reciprocal, but sometimes ਕਈ ਵਾਰ unequal ਵੀ ਹੁੰਦੀ ਹੈ. Frequency of crossing over is closely related to physical distance between the genes. ਕਿੰਨਾ distance ਹੈ between the genes, that is known as frequency of crossing over. It is related to the distance between the genes. Crossing over is very important or crucial process that generates genetic difference within the population. Now what are the significance of crossing over? Crossing over provides strong proof in favor of linear arrangement of the genes on the chromosome. So ਇਸ ਤੋਂ ਸਾਨੂੰ ਪਤਾ ਲੱਗ ਰਿਹਾ ਹੈ ਕਿ ਜਿਹੜੀ crossing over ਹੈ gives a proof in favor ਕਿ ਜਿਹੜੀ genes ਦੀ arrangement ਹੈ on the chromosome that is in linear manner. Recombinations ਜਾਂ ਫਿਰ new gene combinations are produced by crossing over. So which will lead to changes in the genetic pool. So it will lead to evolution. Crossing over generates genetic difference within the population. It also helps to construct linkage maps on the basis of crossing over. Linkage maps बनाए जा सकते हैं. Now what are linkage maps? Maps ਜਿਹੜੇ ਹੈ that are also known as chromosomal maps ਵੀ ਕਹਿ ਸਕਦੇ हैं ਇਹਨੂੰ. They show the sequence of genes on the chromosomes. Genes ਜਿਹੜੀਆਂ further apart have a greater recombination frequency. So linkage of genes in a chromosomes ਅਸੀਂ represent ਕਰ ਸਕਦੇ ਹਾਂ in the form of genetic maps or linkage maps ਕਹਿ ਲਓ, ਜਾਂ ਫਿਰ chromosomal maps ਕਹਿ ਸਕਦੇ ਹੋ। First chromosomal map was given by Alfred Strutvent in Drosophila melanogaster using gene recombination frequency. So recombination frequency is equal to total number of recombinants कितने बने हैं divided by total number of progeny into 100, that will give the amount of recombination frequency. So features of genetic recombination, gene mapping ਜਿਹੜੀ ਹੈ ਸਾਨੂੰ help ਕਰਦੀ ਹੈ to know the order of genes and relative distance between them. ਜਿਹੜਾ ਡਿਸਟੈਂਸ ਹੈ ਅਸੀਂ ਉਹਨੂੰ ਮੈਪ ਯੂਨਿਟਸ ਵਿੱਚ ਦੇ ਸਕਦੇ ਹਾਂ। ਇੱਕ ਮੈਪ ਯੂਨਿਟ ਇਜ਼ ਇਕਲ ਟੂ 1 cm. cm means centimorgan after name of the scientist Morgan. Gene mapping ਜਿਹੜਾ ਮੈਥਡ ਹੈ helps to method is used to find recombination frequency between the alleles in order to find the relative distance between them. Recombination frequencies between the genes are inversely proportional to their distance. So distance measurement is equal to 1 map unit. That means 1% of recombination. Now let us summarize our topic, that was crossing over and linkage, and let us see the difference between the two. Crossing over means crossing over leads to separation of the genes, of the linked genes, and linkage, it keeps the genes together. Crossing over involves exchange of segments between the non-sister chromatids, whereas linkage involves the individual chromosome. In case of crossing over, frequency of crossing over can never exceed 50%. 50% parental combination and 50% ਜਿਹੜੀ ਹੈ that is the recombinants. But in case of linkage, the number of linkage groups can never be more than haploid chromosome number. Crossing over increases variability by forming new gene combinations. Linkage, it reduces variability. Crossing over, it provides equal frequency of parental and recombinant types in test cross progeny. But in case of linkage, it provides higher frequency of parental types than recombinants in a test cross progeny. So linkage retains the parental characters, whereas crossing over leads to new recombinations. Thank you.